we close the discussion about how we study the microorganism in the environment, let us go further quickly into few different types of the marine microbes. So what I will do here is I will explain you about a little bit about marine bacteria, about marine archaea, about marine viruses, right? So quickly have a look. I mean, we will have a look into bacteria, archaea, and viruses. So marine bacteria, it can be any bacteria can be of two type based on the staining technique, right? We know it can be either gram positive or it can be either gram negative, right? Based on the gram staining, right? For any bacteria, we provide a binomial system of Latin name. How you have seen for the animals that there is a binomial nomenclature, it means that there is a genus name and then there is a species name, right? Similarly, for bacteria also, we provide genus and species name. Okay. In order to identify any bacteria or in order to find out the classification of the bacteria, we have to perform polyphasic taxonomy. So a couple of slides back also, yesterday I told you what polyphasic taxonomy means. It means that integration of various different kind of methods. For example, chemotaxis, chemotaxonomy, that is utilization of the cell membrane carbon, right, fatty acids as a proxy for species identification, or molecular method like 16S rRNA gene phylogeny. Okay, so this was mostly done in the past. Even up to close to 10 years back, we could do that, and then we can name a bacterial species, right? Many of us who are from the life science background may have heard about the Burgess manual. So Burgess manual is a kind of systematics of for the systematics of archaea and bacteria, telling us that how if what all physiological uh, features of the bacteria tells us, and according to that we can identify it to whether it is a vibrio, whether it is a salmonella, or whether it is a selenovibrio, and so on, right? However, in coming days we have mostly utilizing genome wide phylogeny, and as a result of which a number of previously known bacteria has been reassigned the name. Okay, and as you can see that in NCBI database, which I yesterday showed you, has 73% of the genome currently assigned to a taxon had one or more changes. It means that previously what it was known as a different name, after doing the genome analysis, it has been shifted to a different clade or different uh, uh, systematic position. Okay. For bacteria, we do not have any formal system of classification, but then there is a rule which is made by International Committee or Systematics of Prokaryote. To that, for the classification of prokaryotes, previously it was known as International Committee for International Union for Bacteriological Nomenclature, which is now renamed as International Committee on Systematics of Prokaryote. And it provides a full set of rules and regulations before you can name a species. However, one of the condition in ICSP is that you have to publish any new bacteria in this particular journal. Until unless you publish in this particular journal, either as a description or otherwise you can describe anywhere else, but you can put in the validation list in this particular journal, International Journal of Systematics and Evolutionary Biology. Fine. So what we have understood, we have understood that in case of bacteria, uh, if we have to name any species, or if we are working on a bacteria and if we have to identify them, we have to follow ICSP nomenclature rules. And then one of the rule is that you have to publish it in International Journal of Systematics, right, and Evolution. in order to publish in that particular journal, one of the idea is to do the whole genome sequencing, because as I said, that until unless you do the whole genome sequence these days, it is not clear that what your species is. Based on the genome you can identify the species. I will restrict that there by saying that we need to require genome these days in order to identify any bacterial species. Okay. Now that we know that there is a molecular era in microbial ecology, right? So there are two terms which has been evolved in last 10 years. First is the rare biosphere. So rare biosphere by mean you can understand that those organisms which are present in a very small quantity. Okay. It means, suppose if there is a species of bacteria, right, then they are present in a very less number of individuals. Okay. Likewise, it is the same for fungi, for archaea, or anything. So in this graph you can understand that there are percent sequence difference from the closest known bacteria. All this group from here to here, you have the species, okay, which are represented by very less number of sequences. So it means that, suppose in this place, let us suppose we say species A. So species A do have a very less, let's say 10 or 20 sequence only, right? So in case of molecular biology, when we are using molecular biology for microbial ecology, it is very difficult to say in terms of a species. So we have to restrict ourselves to tell that what all different taxa are there. And so taxa in this in here means that, suppose if you have a species A and then if you have a species B, and here if the gene or the genome of a species A to the species B, if it is 100% similar, then both the individual species, I mean individual A and individual B, what I said, species A and species B, belong to the same species, right? So if it is more than 94% similarity, right, then it is coming under the same genus. If it is more than 95%, 97% similarity, then it is the same species. If it is 86% similar, then it can be the same family. If it is 82% similar, it can be same order, right? So likewise, you can see that if it is 75%, so all the phylum in the bacteria, if it is belonging to more than 75% sequence similarity, it can, you can call it as all coming under the one phylum. Now to make these things little clear, I will remind you the example which was taught by Prakash, that he took one sea cow and one, I guess, penguin and one, I guess, dolphin, right? So all three were sharing some certain characters up to a certain taxa, up to a certain level, right? That's why they all were coming in one particular group, like let's say mammalia. But then as you go as you go further, then they all belongs to three different species. Similarly, you can understand that if we have a species, if you have any individual A and any individual B, right? Now, between A and B, if they have more than 75% similarity at the genome level, then we can say that A and B both belong to same phylum. Okay. Now, after 75, if they have more than 78, it is same class. More than 82, it is same order. More than 86, then it is same family. More than 95, then it is same genus. And if it is having more than 97% similarity, it is the same species, right? If they are having more than 95% similarity but less than 97% similarity, they could be one genus, but then they belong to two different species. This particular concept is called as operational taxonomic unit. Okay, because just with the genome, without considering the morphological and the meristic characters, you cannot specify it into any species level. Okay, so you can also think it in the form of MOTU, that is molecular operational taxonomic unit, right? And So for example, here I am giving you one example from my paper, which was published for nematodes quite, I mean, close to 5 6 years back. You can see that there are two locations in Maharashtra from where the soil sediment was taken, soil sample was taken. In one particular sample, you had number of OTU observed 10, close to 10, let's say 12, okay, and then one close to 25, okay. So what it tells you? It tells you that in the sample which was taken from agar, okay, you have 41 sequences belonging to 13 different taxa. So since I am not saying a species level, I am just saying that there could be 13 taxa in overall 41 sequences what I have analyzed. Similarly, in Karel, you have 23 different taxa from 71 sequences which I have analyzed. Okay, so you can say that if you, I don't remember exactly that how much difference I took, maybe 3%. So you can say that there could be 23 species of nematode in one location and 13 different species in the second location. Okay, so this is how you understand operational taxonomic unit. And wherever you have a very less number of sequences in each OTUs, that is called as rare biosphere. Okay, it's a very recent term. It is coined after 2010. So now you can understand that how recent these changes are. Okay, let us go back. Maybe we will use these terms in the coming slides. That's why I introduced you here. Now, we know that there are bacteria everywhere in the sea, right? We said that it could be gram positive, it could be gram negative, and we also said that the nomenclature of the bacteria depends on the terms and conditions made by International Committee for Prokaryotes, right? Systematics of prokaryotes. And then we have to perform the genome sequence in order to identify the species. So having said that, that is mostly if you have the cultivated diversity, right? Then only you can go with the genome, single species genome. Uh, but however, we know that the uncultivated diversity is much more, right? And for that, since we can't go up to the species level in uncultivated diversity, we went with the OTU, that is operational taxonomic unit, and we say that based on the OTU, we can define a kind of a species for the uncultivated bacteria, right? Whenever we have to find out the diversity and distribution of marine bacteria, or for, let's say, for any diversity study, we have three different terms, right? Alpha, we have beta, and then we have gamma, right? So alpha can be understand that within the local site or within one particular site. Beta diversity, which is when you are comparing site 1, site 2, and site 3, and whatever is whatever the diversity is, right, that is beta. And for the whole landscape or seascape, if you want to know the diversity, that is the gamma diversity, right? Even yesterday I was telling you that there is a variation in the species composition or the diversity, right, within the site, mostly because of the adaptation in the different conditions. So in the same site you can have a depth profiling, right, where you have the photic zone, and then where you have the aphotic zone. And because of the depth variability, you can have the species composition different. Some will be adapted to the light, some will be adapted to the dark, right? Some of them could be phototrophs, some of them could be heterotroph, some of them could be chemolithotroph or chemoautotroph, or various different metabolic diversity will be there, right? Then some of them will be free living directly into the water column, and some will prefer to attach on the either on the phytoplankton, zooplankton, or otherwise also on the dissolved organic matters through some appendages, right? Or some will be what do you say, free living. At the same time, we also have a very patchy distribution, because even in one particular site, just imagine that you have a open sea, And then within the open sea, at one place you have more phytoplankton. So this phytoplankton will do, we have seen already that this phytoplankton can excrete the exopolysaccharides, right? Because of those exopolysaccharides, there are bacteria which are specifically attached to that particular area, right? Or you can say that those bacteria could be living very much near to the phytoplankton, right? Similarly, because of all the physical uh, mm, features, change in the current, change in the waves, or maybe also with the vertical migration, we may have a patchy distribution for nutrients, right? And these nutrients can also determine that what kind of diversity will be there. So apart from that, there would be certain bacteria who are syntropic. So syntropic, what it means, that syntropic relation, it means there are, if there are two bacteria, let's say bacteria A and bacteria B. So if bacteria A is producing some nutrient, right, and bacteria B is producing some nutrient, A cannot produce this particular thing, so it will uptake. B cannot produce this thing, so it will uptake, right? So that kind of interactions will happen. So what will happen if A is present there, then B will also be present. If A is not present, then B may not be present there, right? So because of all these interactions, right? And also whether the phages are I will show you also in the latter part of the talk today that how phages regulate the biodiversity of microbes, right? So based on all these factors, there is a high alpha diversity in the bacteria, in the marine bacteria. Okay. Now, one of the major factor which defines the alpha diversity is the vertical structure of water column. And that vertical structure is nothing but in the surface you have the highlight, in the surface you have the warm water, in the surface you have the phytoplankton. As you go down, you will reach a line of thermocline, right? So that thermocline layer changes the changes the conditions in the water column, and that also defines the bacterial diversity in the ocean. Okay. Now also between, I mean, beta diversity also varies. How you can see in this way that there is a, if there is a site 1 and site 2, and site 1 is, let's say, estuary, and site 2 is a marine. So in the estuary you will have the high nutrition content, right? So that defines that will have more copiotrophic marine bacterial diversity, right? Whereas in the marine, if it is oligotrophic water, then there will be a more oligotrophic, extreme oligotrophic microbes. Similarly, from coastal to offshore, it's the same how you can understand estuary to marine, and also in the stratified water column or the ocean front. So in different part of the ocean, we will have different sets of microbes or a very wide range of diversity because of the varying abiotic and the biotic factors. Okay. In 2004, I guess, there was a census, international census of marine microbes that was made uh, in order to understand marine microbes of the open ocean or of the marine environment, right? So they have studied a lot about different alpha, beta, and gamma diversity of the marine bacteria, right? We will see that what are there some of the results. Okay, you can see in this particular slide what is that what I was telling you here, that there could be a variation in the bacterial composition, which indeed you can see that a paper which was published in 2011, that there is a more and more of alpha, gamma, and the alpha bacteria, alpha and gamma proteobacteria, and flavobacteria in the coastal waters. Okay. Whereas you can see that all these bacteria, let us suppose we will look into epsilon proteobacteria. So this epsilon proteobacteria is mostly present near to the vents, right? And then you also have some of the, so alpha proteobacteria, gamma proteobacteria will be one of the most I mean they are the one of the most abundant bacteria in the marine environment. So I don't know if I have mentioned here, but no, I have not. But what happens is that almost half of the marine bacteria is gram negative bacteria, and that belongs to alpha and gamma proteobacteria. Okay, so you can see that bacterial community composition in different reams ecosystem types changes. Okay, and that result has came from international census of marine microbes, which I said that was made in 2004, and they have published a number of articles in this aspect from 2010 onwards. Okay, you can see that in most of the pelagic water you have alpha and the gamma proteobacteria followed by flavobacteria, and then in this order. Similarly in the benthic, so I guess you remember what is pelagic and what is benthic now. So pelagic is a water column, and benthic is the sediments, right? So in sediment again you can see that the gamma proteobacteria, delta proteobacteria, and alpha proteobacteria are abundant, followed by other different groups of bacteria, right? And within alpha proteobacteria, you have a group of bacteria called as SAR11, which I have been telling you since yesterday. That was named because it was uncultured at the time of the name was given. That is Sargasso C. That's why it is called as SAR11. And now it is more than 20% of the total bacteria in the marine environment, right? So I have given what it means in this graph in this particular paragraph, but overall what you need to understand that there is a changes in the bacterial composition from coastal water to surface water to deep to anoxic to vents to so this is pelagic vents, this is benthic vents, this is deep sea, and then again this is a benthic coastal sites, right? So that is how it is again proven that the bacterial uh, that bacteria is having high alpha and beta diversity. Now, if you look into the global picture of the marine bacteria, what you will find is that it's the same result, that gamma and the alpha proteobacteria in all the sea, be it a Southern Ocean, Indian Ocean, Atlantic Ocean, or wherever you will see, right? You see that this brown and this blue, these two, right? Of course, there are local variations. For example, here you have more bacteroids, here you have more bacteroids, right? But overall, what you see is that gamma and alpha proteobacteria are abundant in across the sea. And in particular, alpha proteobacteria, which belongs to 14 different orders, 15 different families, and altogether there are more than 400 species which could be cultured till date in the plates. Okay, and this includes your SAR11 and SAR116. And as I said, that proteobacteria is the most abundant and widely distributed in all major habitats. So in future, if you are ever working on marine microbes, there are more chances that you will get proteobacteria as an abundant bacteria in your cultured plate. Okay, so gram negative bacteria is the most abundant, and because these gram negative bacteria are having a very high metabolic diversity, which we have learned by now that defines the adaptation in the different habitats. So if you have a high metabolic diversity, then there are chances that you will survive in a wide varieties of habitats, right? So this graph is taken from Pomier et al 2007. However, there are recent paper as well, which is published in 2020 in the journal Cell, okay, and which again tells you about the global trends in the marine planktonic diversity based on the different latitudes across the world ocean, okay, and you can see prokaryotic diversity mostly for bacteria. Uh, P is photosynthetic, H is heterotroph. Okay, so likewise you can see the dominance relative abundance in the different latitude, right? So if you are interested, you can just go and have a look into this particular new paper 2020 paper. Okay, Now that we have seen that the bacteria are present, I mean, it is having a high alpha and beta diversity, and of course, globally, you have a huge biodiversity and mostly dominated by gram negative bacteria, in particular to proteobacteria, and that also alpha proteobacteria. And I have given you the detail that there are chances if you are working in the marine microbes, you will get alpha proteobacteria in higher number in the open, I mean, in the marine environment. Let us see some of the special bacteria, for example, which performs unique movement. This is a magnetotactic movement, which is shown by some of the bacteria belongs to alpha proteobacteria, and then there are some related, I mean, some bacteria are also there in delta and gamma proteobacteria. These bacteria do have a magnetosome, okay, magnetosomes inside the cell, and these magnetosomes will import a lot of magnetite and the greigite particles from the environment. So how do they do it? Again, using the siderophores. So yesterday we have seen that how do they acquire the iron? They acquire the iron through the siderophores, and then they have the specific receptors on the cytoplasmic membrane. So these bacteria do do uptake a lot of magnetite and the greigite ions, and as a result of which they develop a kind of magnetic behavior, and they orient themselves in the earth's magnetic field. Okay, after that they use the flagella in order to come towards the light. Okay, so that is how this magnetotactic bacteria are there. You can also cultivate them if you take from the sediments or from the salt marshes bacteria, and then you use a magnet near to the petri plate or near to the test tube. There are chances that you will be able to isolate this magnetotactic bacteria, which is mostly belonging to this magnetospira and magnetospirillum. Okay, but it is very even though if you will able to isolate, it is very difficult to cultivate it. You can see in this particular image that this is how a magnetosome will look like, where you have the magnets, I mean not magnets, this iron particle uh, which will align themselves according to the earth's magnetic field. Okay, so this is a special group of bacteria in the marine environment, right? Now there is another bacteria, though there are many and we cannot cover into our all the stories of bacteria, but I will just tell you some of the most exciting ones. The family Vibrionaceae, right, which is a Vibrio, and this Vibrio includes many pathogens. So maybe I will just briefly touch upon today even the pathogens, but mostly we will be learning this in the class of fish and fisheries, where we will deal mostly on. I mean, we will also deal about the pathogens of the fishes. But what we need to understand right now is that many of the Vibrio species are causing diseases or are pathogens to many fishes. For example, Vibrio angularium, you have photobacteriums. It can cause disease to mollusks. It can cause diseases to a lot of crustaceans by Vibrio harvae, and so and so on, right? So they are also very opportunistic pathogens. So so if there is any compromise in the health of the corals or any of these marine organisms, this Vibrio will be first to attack them. Okay, in the Vibrio, their peculiarity is that they have close to 40% bigger genome size than their comparable marine bacteria. As a result of which, they have a number of genes in them which causes virulence. Okay, and these virulence genes are causing the, I mean, acting as a pathogens to the many of the marine organisms. Okay. Now, in the same Vibrio, we do have a special property called as bioluminescence. So many of the bioluminescent bacteria belong to the family Vibrionaceae, two examples. One I have already given you, this particular example where you have the symbiotic association with the anglerfish in the deep sea, which provides bioluminescence to the fish in the esca of the fish, that attracts the prey, that attracts the mate, right? So all those functions have been done by this particular group of bacteria in the anglerfish. Also, you have a lot of vibrio in on the skins of the squids and other cephalopods, which are also causing the bioluminescent in the night time. Okay. This is also a very peculiar to the Vibrio, that they have close to 1600 core gene. So what core and pan means is that, suppose if you have 30 different species within Vibrio, then 1600 genes are core to all the 30 Vibrio. Whereas if you add species A, suppose having 4000 gene, species B suppose having 6000 gene, likewise they have 22,000 genes. So out of 22,000 gene, only 1630 are core gene. So this core gene will be responsible for a common behavior across all the Vibrio, but the unique gene to each species can lead to different virulence, different pathogenicity. That's why in the family Vibrionaceae we have a lot of pathogens. Okay. Now, another peculiar activity is the bioluminescence, and we know that bioluminescence, it's a kind of a collective effort of all the Vibrio species in one particular location. So, if the cell density is lower, that means if there are very less number of cells of Vibrio in one particular location, it will not cause the, or it will not activate the luxoperon, which is responsible for the bioluminescence. If the number are increasing, and number increases because of the auto inducers, again not going in detail, but just understand that the Vibrio releases the inducers in the atmosphere, I mean in the vicinity. Sensing that inducer, more and more Vibrio will come. Now once more and more Vibrio will come, then again this auto inducer will uh, induce the translation of, or the whole operon will get activated, that lux operon, and that will lead to the bioluminescence. So it is very much important for a high cell density for the bioluminescence. And also there are some concept like a squirm sensing, that is a communication among the bacteria, that is also depending on these auto inducers releasing in the environment. And this quorum sensing also leads to the biofilm formation, which I was telling you in the last class, right? So what you need to just understand is that this Vibrio is a very good model to study the bioluminescence, to study the quorum sensing, and to study the biofilm, right? Because these all factors are very much connected to each other. And also you need to remember that bioluminescence, one of the factor that bioluminescence was evolved in order to protect themselves from the from, I mean, to repair the damage for the photolysis, right? So all those things are very much interconnected, which we have been talking in the last 2 3 days. Bacteria are not only symbiotically associated with the animals but also with the plant. This particular example is a very fascinating example. And when I visited very first time this zoological station in Roscoff in France, there I met a scientist called as Simon Detami. So he was doing his PhD on this particular aspect, that in order to understand the role of bacteria in the adaptation of seaweeds. So there he explained that, and now that he has published several articles regarding this, what is being I mean happening is that if there are certain bacteria associated with the ectocarpus, which is a brown algae, right? And if that bacteria is associated with the ectocarpus, brown algae, then the bacteria is capable to grow in a fresh water medium. If he is using the antibiotics and removing those bacteria, then that particular bacteria is, since it is not there, it is not releasing the hormones required by the ectocarpus in order to grow in the low salinity water or freshwater medium, right? So this is how such importance is there for the bacteria in the host microbiome interactions, which can actually derives the evolution or the adaptation of the marine organisms in the different environmental gradients or different abiotic conditions. Okay, right? So you can see the whole experiment coming out from this particular paper. It's a nature group paper, again a very high impact paper, where you can see that once he is removing the antibiotic treatment, putting in the fresh seawater medium, then you can see a good growth of the algae, right? And that is directly linked with the photophysiology of the algae, which is measured using the instrument which I will be teaching you next week. Okay. Apart from that, there is the same research group in collaboration. So in Europe, this is a multi-million euro project going on between 17 different research groups. They have found that the bacteria is also involved in the proper morphological development of the most common seaweed, that is ulva. What is happening that there are two group of bacteria, that is, two species. One is rosiobacter, another one is cytophaga. So when the rosiobacter and cytophages are removed from the medium, then this particular seaweed is forming a callus like structure, as you can see here, right, in this. So it is not able to make a particular shape. However, when they are reinserting this rosiobacter separately, so it is promoting the cell division. So it means that rosiobacter is giving some certain regulators for the cell division in ulva. And then the cytophaga, that cytophage, when it is added back to the ulva, it is leading to the rhizoid formation. So you can see the rhizoids here, right? Only once you combine the rosiobacter and cytophaga, then a proper structure of the seaweeds is coming. It means that this macroalgal microbiome, that includes the rosiobacter and the cytophaga, helps the ulva, or helps, let's say, any other seaweeds as well, to take a proper cell morphology. Okay, if you are removing that, then the morphogenesis of the ulva will be infected or the affected, right? And right, uh, I will stop the bacteria after this example, which came in 2010 in Nature. Now, how this bacteria is modulating our gut microbiota, human gut microbiota, can be very well understand with this particular example. That since Japanese people are eating sushi, right, for a lot of time, for a number of generations, right? But we, as a human, inside the in our gut, we do not have the G or the enzymes which can uh, break down this complex carbohydrates, right? That is porphyrinase. This particular enzyme was found in the gut of the Japanese people. Okay, so there, and then when they looked into the similar enzyme from the gut of North American population, I hope I have written somewhere. Yeah, in the North American population, it was not there, because most probably in North Americans, they are not eating sushi that frequently. How the Japanese people are eating, right? Now, this in the Japanese people gut, there could be a horizontal gene transfer. Okay, because of the bacteria present on the surface of this particular seaweed. And this particular seaweed, when the Japanese people might have eaten raw, the bacteria associated with it could have given the through the horizontal gene transfer to the bacteria living in the gut of those people, right? So you can see that there is a bacteria on the seaweed surface, which after ingestion might have given through the horizontal gene transfer the gene responsible to digest this complex carbohydrate to the bacteria living in the gut of the Japanese population. So it came as a very interesting finding, the transfer of the carbohydrate activated enzyme from marine bacteria to Japanese gut microbiota. And this also gives a very good example that how we still are evolving uh, depending on the marine microbes. Okay. Apart from that, there are several other important marine bacteria are there. One example I gave you in the last week about the DMSP degradation uh, in the sulfur cycle. So there is this particular species called as Oceanospirillum, right? So this Oceanospirillum is involved in the DMSP degradation to DMS in the sulfur cycle. Similarly, we have a lot of different bacteria who are engaged in the degradation of the oil spills and hydrocarbons at the natural volcanic seeps. Okay. And again, we have the cyanobacteria, Prochlorococcus and Synechococcus, who are actively involved in the photosynthesis. And uh, every time we breathe the oxygen, it at least 20% of it is coming from the Prochlorococcus and Synechococcus. Okay. Then we have also a very fascinating bacteria in the marine environment that is called as Vibrio species. So this Vibrio species, you can see that they are very much, I mean, they are predatory bacteria. And as soon as they are in the yellow one is the Vibrio species in this particular image. And then you can see that it infects to the, you see that after the infection, it lyses the cell of the host bacteria and then reproduce and then come out. So this kind of predatory bacteria is also present in the marine environment. One of the good example is the Vibrio species, and Vibrio species is also involved in the pathogenicity of the oysters. Since oyster is one of the delicious food in the sea food, this particular bacteria is also causing harm for the oyster fisheries. Okay.